The cell theory, coined by Theodore Schwann, is composed of three basic principles. 1. All living things are composed of cells, or cell products. 2. The cell is the smallest unit of life. And 3. All cells come from pre-existing cells. All principles of the cell theory have supporting evidence. For one, when viewed under a microscope, all living things have been found to be made up of cells or cell products. Observations made by Robert Hooke, Anthony van Leeuwenhoek, Matthias Schleiden, and Theodore Schwann all support this conclusion. 2. Scientists have yet to find a living entity that is not made up of at least one cell. And 3. Experiments conducted by Louis Pasteur in the 1860s have shown that cells cannot grow in sealed and sterile conditions. Through a sterilized chicken broth experiment, Pasteur showed that living organisms would not spontaneously reappear, and life was only able to re-establish itself after exposure to pre-existing cells. All organisms, whether they are unicellular or multicellular, carry out all of the functions of life. These are 1. Metabolism, which are all the chemical reactions that occur within an organism. 2. Growth. Living things can grow, changing in size or shape. Even if limited, growth is always evident in one way or another. 3. Reproduction. All living organisms are able to produce offspring, either sexually or asexually. 4. Response. Living organisms are able to respond to their environment. 5. Homeostasis, which is the capability to maintain a constant internal environment. This consists of a regulation of temperature, acid-base levels, etc. And lastly, 6. Nutrition, which is an exchange of compounds, such as gases or proteins, with the environment in order for the organism to obtain the energy and nutrients necessary to perform all of its other functions. From smallest to largest, let's compare the size of a few things. A molecule is approximately 1 nanometer long. Cell membrane thickness is around 7.5 nanometers. A virus is approximately 100 nanometers long, with a range of 20 to 200 nanometers. A bacteria has a size ranging from 1 to 5 micrometers. Organelles are greater than that, but still have a size less than 10 micrometers. And eukaryotic cells are greater than organelles, but are smaller than 100 micrometers.
So first we need to calculate the linear magnification of drawings and the actual size of specimens in images of known magnification. So what do we use? We use this formula. Magnification equals size of image divided by size of specimen. So first we need to know the magnifications. And the magnifications that we have in our microscope right here are 10, 20, 50, and 100. Now we also have to know a little bit about the specimens we're trying to know. So basically just have an idea. So molecules are 1 nanometer. And then we have that biological membranes are around 10 nanometers, and then viruses are about 20 to 300 nanometers, which is the biggest range here. And then we have that bacteria are 5 micrometers. It's really different. You have to have that in account. So basically, if they give you all of this, if they're gonna give you magnification, and now you have the idea, then you grab your ruler and actually measure the drawing that they give you, and then you can use the formula and get the answer. It's not that hard. And now we move on to 2.1.6, in which we have to explain the importance of the surface area to volume ratio as a factor of limiting cell size. So why is it important? It happens that if you have a really big surface area, or like a really big cell, the surface area cannot bring in needed materials and get rid of cell waste, or easier okay, never mind. So and also it's easier to have a smaller cell, it's easier to have the functions of life, which is metabolism, growth, reproduction, response, homeostasis, and nutrition. Remember those well. And then we also have that larger animals have more cells, not bigger cells. So imagine a giraffe. Yes, long neck. Ooh. It's not really pretty. But imagine a giraffe. And then you might say like, oh, their cells are ginormous. But it's not like that. We actually all have these kinds of cells right here. Little. And it actually forms the whole giraffe. So that's really important to know. And another thing that cells do in order to have more surface area and less volume ratio is to have infoldings or outfoldings to increase the surface area. So this happens in our body. We can see it with villi in the small intestine. So I'll try. Oh goodness. So here we have the stomach. Okay, don't do this in your test because it's clearly not gonna work. And then like right here, if you zoom in, then you have the infoldings. So what happens is that we actually remember that we don't have the like blocks or anything that can like wait in order for the food to be digested. So then we have little like hair-like things in our beautiful intestine in order to make more surface area and have more and grab more nutrients. So another thing that you really need to remember is that multicellular organisms show emergent properties. And this is our beautiful 2.1.7. And with this, we go to another person.
The next section asks you to explain that cells in multicellular organisms differentiate to carry out specialized functions by expressing some of their genes but not others. This process of differentiation will occur when certain DNA segments are activated by chemical signals, which will result in the expression of certain genes but not others, and will allow cells to produce all the required cell types, in this way becoming specialized. Section 2.1.9 asks you to state that stem cells retain the capacity to divide and have the ability to differentiate along different pathways, in this way having the possibility to differentiate into any other cell in the body. The last section asks you to outline one therapeutic use of stem cells, and I have selected a couple examples for you to choose from. The first two examples are a part of the therapeutic cloning, which is when stem cells are grown in order to replace differentiated cells lost during disease or injury. The first example would be Parkinson's and Alzheimer's disease, which are both caused by loss of brain cells. The treatment would be the implementation of stem cells that would hopefully replace these lost brain cells, in this way relieving the symptoms. The second example would be diabetes, because there are certain forms of the disease that deplete the pancreas of essential cells, and the treatment would hopefully implement stem cells that will have a positive effect on these lost cells. The final example is part of the tissue-specific stem cells, which are stem cells that are found in certain tissue types and that can only produce new cells of that same tissue. The example would be leukemia, which is cancer of blood-forming cells. The treatment of this disease would be blood stem cells being induced in patients in order to replace the damaged bone marrow. The final topic to be discussed would be ethical issues surrounding the use of stem cells. The main issue would be surrounding the use of embryonic stem cells, which come from embryos, and in order to be gathered, the death of the embryo must take place. Some argue that killing would represent the taking of human life. Nevertheless, others argue that the research could really result in reduction of human suffering, and they therefore believe it should continue.